Next chapter, the cell division. Now, we learned about the structure of the cell and its functions. We know that all living organisms are made up of cells, which are the basic functional units of all living organisms. When an organism is born, it already has millions of cells that make up its body. But perhaps what is not so obvious is that life begins as a single cell, a fertilized egg which we call a zygote. Then through the process of cell division, the organism increases in size as well as complexity until maturity. Cell division is basically the splitting of a cell into two new daughter cells. Cell division is only a small portion of what is generally called the cell cycle. From this cycle, you will see that the cell spends much of its time in the region called interphase. During this time, the cell is preparing itself for the process of cell division. For most organisms, the cell cycle takes 24 hours and cell division is only a mere 80 minutes event. So what exactly happens during interphase? If you look at the cell dividing, you will see that the daughter cells have all the organelles that the cell needs to carry out its functions. These include the mitochondria, Golgi bodies, rough ER, and so on. So before a parent cell divides, it must double the number of each organelles so that each daughter cell will have adequate organelles to be able to function. The most important of them all is the nuclear material, which is basically the cell DNA. DNA is the control center of the cell, and without it, the cell can't exist. So cell division also involves the division of the cell DNA into two for each of the daughter cells. This is the main focus of this chapter, nuclear division. To be able to understand this division, let us first understand some terminologies: DNA, chromosomes, genes, and chromatids. We said that the nucleus is the control center of the cell. It contains the DNA, which is the genetic material. The DNA contains all the genetic information about the organism. The DNA is not usually one long continuous strand. No. It is split into numerous strands of varying lengths. In a non-dividing cell, the DNA exists as tangled spread-out threads called chromatin. When a cell is ready to divide, the DNA condenses into structures called chromosomes. The chromosomes can be visualized using a light microscope. The chromosomes are the structures that will be involved during nuclear division. Next is chromosomes. Chromosomes are the condensed form of the DNA. Different organisms have different numbers of chromosomes. But all body cells of an organism have the same number and type of chromosomes. Some sections of the DNA are called genes. These are the coding regions and determine the features of an organism, like height, hair color, color vision, and color of flowers in plants, and so on and so on. The position of a gene in a chromosome is called its locus. In the body cells, chromosomes occur in pairs. Each pair of chromosomes has a characteristic length. The member of each pair is called a homologous chromosome. Homologous chromosomes are similar but not necessarily identical. This means that they contain the same type of genes, but the genes do not necessarily code for identical characteristics. Let's explain that with an illustration. Take an example of a cell with only two chromosomes. Each has a homologous pair, and you see that the homologous pair are the same size. Imagine the colored region represents genes. For the homologous pairs, you see that the genes take up the same locations on the chromosomes, that is their loci. So what does it mean to be similar but not identical? Let's use the first homologous pair as an example. The chromosomes have 3 genes each. Let's say these genes code for height, these ones code for hair color, and these ones for eye color. But although these ones both code for height, one can be for tallness and the other for dwarfness. You see, they both code for the same characteristic, height, but the alternative forms of height. We call such pair of genes alleles, the alternative forms of the same gene. So tall and dwarf are the alternative forms of height. For the hair color genes, one may code for black hair and the other for brown hair, but both code for the same characteristic, hair color. But then they don't always have to be different. The third pair of alleles may both code for brown eyes. So where do homologous chromosomes come from? You get them from your parents, one from your father and the other from your mother. When a cell has both members of the homologous pair, it is said to have a diploid of chromosomes. This is also called the 2N constitution. All body cells have a diploid of chromosomes. If a cell has only one member of the homologous pair, it is said to have a haploid of chromosomes. This is also called the N constitution. Sex cells like sperms and ova have a haploid of chromosomes. During fertilization, they fuse to form a diploid zygote. And that is how you get the homologous pairs of chromosomes. And so, as the cell duplicates all the other organelles, it must also duplicate the chromosomes during interphase. So each chromosome is replicated, but the two remain attached to each other by a structure called the centromere. Each arm of the duplicated chromosome is called a chromatid. The two chromatids joined by a centromere are called sister chromatids. They are identical since they are copies of each other. So when you see a chromosome with an X shape, then it means it has undergone the interphase stage. If you look at the human body cells, there are 46 chromosomes, and each chromosome has a homologous partner. This means there are 23 pairs of homologous chromosomes. In this case, the cell has not yet undergone DNA replication or the interphase. So we say that we have 46 non-replicated chromosomes, which is basically 23 non-replicated pairs of homologous chromosomes. Homologous pairs only exist in body cells, which are called somatic cells, and not in sex cells. Sex cells only have one member of each homologous pair, so they have half the number of chromosomes as the body cells. Homologous pairs of chromosomes have the same size, carry the same genes, and the genes occupy the same loci, except the X and Y chromosomes. They can have different alleles and are not held together by a centromere. During interphase, the cell undergoes DNA replication, and each chromosome now consists of two arms called sister chromatids held together by a centromere. Sister chromatids only exist after DNA replication. They are 100% identical, means they have the same size, carry the same genes in the same locus, and importantly, they have the same alleles for the same genes. After all, they are copies of each other. These sister chromatids are collectively known as replicated chromosomes. So now we have 46 replicated chromosomes, which is 23 replicated pairs of homologous chromosomes. Now, if you understood up to that point, it will be easy to understand cell division, and specifically how these chromosomes are separated. There are two types of cell divisions, mitosis and meiosis. Mitosis is a type of cell division in which a diploid parent cell splits once into two diploid daughter cells. Meiosis is a type of cell division in which a diploid parent cell divides twice into four haploid daughter cells. Mitosis. Mitotic division occurs in body cells or somatic cells and leads to growth of an organism by increasing the number of cells. It is also the cell division that occurs during binary fission. During mitosis, the parent cell divides into two daughter cells, each having the same number of chromosomes as the parent cell. It is the process of dividing the replicated DNA of the cell into two nuclei. Therefore, the key feature of mitosis is the preservation of the parental chromosome constitution. Mitotic cell division is a part of the larger cell cycle, taking about 80 minutes. As we earlier said, the cell spends a larger part of its time in interphase stage. For convenience, mitosis is divided into several stages: prophase, metaphase, anaphase, and telophase. These stages are not distinct, but occur in a smooth continuous sequence so that each stage merges into the next and the events often overlap. It is important to note that the cell undergoes mitosis to form two nuclei and then divides into two individual cells by the process called cytokinesis. Interphase is the preparation stage for division. During this stage, the cell grows into nearly its full size and performs all its biochemical activities as usual. The cell also replicates its DNA so that we have sister chromatids. At this point, they are not visible by a light microscope. The cell also synthesizes new organelles like mitochondria, Golgi bodies, and importantly, centrosomes in animal cells. It also builds up sufficient energy in the form of ATP. At the end of interphase, two centrosomes appear on one end of the cell. During prophase, the chromosomes condense and thicken, and therefore they become visible. The centrosomes start producing thread-like structures called microtubules, which will form the spindle fibers. The nucleolus disappears as the nuclear membrane begins to break down. At the same time, the centrosomes move to the opposite sides while microtubules continue to grow. During metaphase, the spindle fibers lengthen and attach to the centromere. The chromosomes align themselves at the center of the cell, with the sister chromatids facing the opposite sides of the cell. During anaphase, sister chromatids separate at the centromere due to shortening of the spindle fibers. The chromatids move to the opposite poles of the cell, and the spindle fibers begin to disappear. Telophase is the final stage of mitosis. At this stage, the components of the new cells begin to appear. The spindle fibers are completely broken down. The chromosomes uncoil and return to an uncondensed state. A new nuclear membrane surrounds each set of the chromosomes at the poles of each cell. Mitosis is now complete. The cell now undergoes a process called cytokinesis in order to divide the cytoplasm into two nearly equal halves. All the organelles, having been duplicated, are now divided between the two new cells. Once formed, the two cells can now enter into interphase to prepare for mitosis, and the cycle continues. In that case, Each daughter cell becomes a parent cell ready to divide. Since plant cell walls cannot be constricted by actin fibers, vesicles form an expanding membrane partition called the cell plate. Like animal cells, plant cells use cytokinesis to finish the division of the contents of the cytoplasm between the two identical daughter cells. And so the key things to note in mitosis is that it involves a single nuclear division. The diploid nucleus divides into two diploid nuclei, and therefore there is preservation of the chromosome number. It occurs in somatic cells and not in sex cells. Mitosis is the basis of growth of a zygote up to a fully grown organism. It is also the basis of asexual reproduction in binary and multiple fission in microorganisms like amoeba. Next is meiosis. Meiosis is a type of cell division in which haploid cells are produced from a diploid cell. It occurs in special cells called germ cells within the gonads of males and females and generates the male and female gametes for reproduction. In order to reduce the chromosome number by half, 2 successive rounds of division are required. These divisions are called meiosis I and meiosis II. Although the processes of meiosis and mitosis look similar, there are a few differences. The obvious difference is that mitosis results into 2 diploid daughter cells, while meiosis results into 4 haploid daughter cells. Just like mitosis, meiosis begins after the cell has undergone the interphase stage. During interphase, the DNA is replicated, producing 2 copies of each chromosome called sister chromatids. The paired centrioles duplicate and begin extending microtubules. Sister chromatids remain attached at the centromere and condense as the cell enters prophase I of meiosis. Up to this point, the cell looks similar to mitosis, but 2 events occur in meiosis that do not occur in mitosis. The first event occurs during prophase I, when homologous pairs of chromosomes lie side by side in a process called synapsis, forming a tetrad or bivalent. Remember, homologous pairs share similar but not necessarily identical genes. Once this happens, the second event, called crossing over, can occur. During crossing over, a physical exchange of chromosome segments of non-sister chromatids occurs, which increases genetic diversity. Prophase I concludes with the disintegration of the nuclear envelope as the duplicated central pairs move to opposite poles of the cell while extending the spindle fibers. During metaphase I, the spindle fibers are now completely formed. The chromosomes, still associated as bivalents, are attached to the spindle fibers by their kineticals. The chromosomes now align along the metaphase plate such that the homologous chromosomes orient towards the opposite poles. The cell enters anaphase I. In anaphase I, the homologous chromosomes separate and move to the opposite poles. Meiosis I ends with telophase I, when the chromosomes decondense and the nuclear envelope reforms. The cytoplasmic material is separated during cytokinesis, and a cleavage furrow separates the 2 daughter cells. The 2 daughter cells now enter the second meiotic division, or meiosis II. The cell enters into a short interphase without chromosome replication. The centrioles duplicate and begin moving to the opposite poles of each cell while extending the spindle fibers. In prophase II, the sister chromatids condense and the spindle fibers extend and the nuclear envelope disappears. In metaphase 2, the sister chromatids attach to the spindles, with the sister chromatids attached to the opposite poles. The chromosomes align along the metaphase plate. During anaphase 2, sister chromatids are separated and move towards the opposite poles. The process ends in telophase 2 as the chromosomes decondense and the nuclear envelopes reform. Cytokinesis occurs and cleavage furrows separate the two daughter cells into 4 haploid daughter cells. The haploid daughter cells will specialize into either sperms or ova. This will fuse during fertilization to form a zygote and hence restore the diploid number of chromosomes. The zygote will divide by mitosis and grow into a child. So the child will get half of its chromosomes from the mother and half from the father. And so in summary, the goal of mitosis is to create 2 identical cells from the original, the same exact DNA and all the same organelles in each daughter cell. First, the cell has to duplicate its chromosomes, and then these 2 copies separate into 2 new nuclei. The goal of meiosis is to make 4 different cells with half the number of chromosomes as the original cell. In meiosis, things start the same way. The cell has to duplicate its chromosomes. But from here, there are some differences. During prophase 1, the homologous pairs may swap sections of the chromosomes, creating more variations. The homologous pairs split up in meiosis 1. But in meiosis 2, the chromatids separate into single chromosomes. This gives 4 totally different cells with half the normal number of chromosomes. And finally, let us perform an experiment to observe the process of mitosis in onion root tip. We will need forceps, needle, a cover slip, glass slide, filter paper, acetyl alcohol, which is a 1:3 ratio of acetic acid and ethanol, onion, beaker with water, hydrochloric acid, acetocarmine stain, watch glass, blade, compound microscope, and a Bunsen burner. Now, place the onion on a tile and carefully remove the dry roots using a sharp blade. Grow the roots by placing them in a beaker of water for 3 to 6 days. Cut off 2 to 3 centimeters of freshly grown roots and place them on a watch glass. Using a forceps, transfer them into a vial containing the acetyl alcohol. Keep them here for 24 hours. Using a forceps, take 1 root and place it on a clean glass slide. Add a few drops of hydrochloric acid on the root tip, followed by 2 to 3 drops of acetocarmine stain. Warm the slide and its content slightly on a burner. Carefully remove the excess stain using a filter paper. Using a blade, cut the comparatively more stained portions of the root and retain it on the glass slide. Then pour 2 to 3 drops of water on the root tip and mount a glass slide using a needle. Now slowly tap the cover slip so that the meristematic tissue of the root tip below the cover slip is properly squashed and spread as a thin layer of cells. Now place the slide on a microscope and observe the different stages of mitosis. By looking at the behavior of the chromosomes, you can be able to determine the stages of mitosis in the different cells, that is interphase, prophase, metaphase, anaphase, and telophase.